How do you look for problematic host cell proteins in your adeno-associated virus-based gene therapy? In this case, a CMO used mass spectrometry to investigate batch-to-batch consistency and identify HCPs of potential concern in 2 AAV products. The products are expressed by infecting SF9 cells with baculovirus. The nature of the product made it difficult to analyze the HCP with commercial ELISAs. Therefore, we set up an LC-MS-based assay to identify and quantify all host cell proteins. Next, our team compared the list of identified proteins to those found in other AAV products and also more traditional biologics. Among the detected HCPs were 2 of potential concern: an isomerase often seen in CHO-produced biologics and ubiquitin, a degradation pathway protein, which we found from both the virus and the SF9 cells. Since LC-MS can detect any protein regardless of the species, we used the same dataset to quantify the viral proteins. In this figure, you see the total viral protein amount in 3 batches of each of the 2 AAV products. The overall viral protein amount was consistent in AAV product 1. However, for product 2, 1 batch contained more than twice the amount compared to the 2 other batches. The client investigated the batch to optimize the manufacturing process. The result was a successful scale-up to 2,000 liter batches without significant differences in the total viral amount produced.